the previous video, we discussed about the nervous system and its different parts, its divisions, and the cells that make up the nervous system. So now we will continue on with the discussion on the action potentials. So remember that the nervous system is like a circuit, as we already said. We always compare it to an electronic circuit because there are electrical impulses passing through the neurons. And actually, that's one of the reasons why neurons are a very special type of tissue, because they have this ability to transmit electrical impulses. But how do they actually transmit those electrical impulses? So the mechanism is because of a membrane potential. So a membrane potential is the result of the difference in the ion concentration between two sides of your cell membrane. So remember, your cell membrane, it's a very thin, it's a hydrophobic. It has a hydrophobic layer. The interior of the membrane itself is hydrophobic. So you have a clear separation of the inside and the outside. So, meaning to say, remember from your chemistry, like dissolves like. So, oil cannot be dissolved in water. So, hydrophobic are, basically, oils are hydrophobic. So, basically, you have a layer that clearly separates basically the membrane that separates two aqueous layer. And what are those two aqueous layer? The interior of the cell and the exterior of the cell. So basically, those are the two sides, the interior and the exterior of the cell. That's the two sides of the cell membrane. Now, a membrane potential results because you have an excess of a charge from one side. For example, the interior of the cell is mostly negatively charged, while the exterior, the outside, is mostly positively charged. And because the cell membrane is hydrophobic hates charged particles. They hate polar particles. Those charged and polar particles cannot pass through the membrane. And so you have basically a fence. The membrane serves as a fence that separates the positively charged exterior to the negatively charged interior. And you have here that unequal charge distribution cause some what you call the potential. It's called a potential because there's a driving force. Because the positively charged ions wanted to go in the cell, the negatively charged ions wanted to go out. Because again, because remember, we are thinking here about ions. There are many negative ions. Of course, they are being repelled by each other. There are many positive ions. They are repelling one another. So of course, they want to move to the other side. But again, because of the hydrophobic layer, they cannot. And that is the membrane potential. So, the membrane potential of a neuron during a typical nerve impulse alternates between negative 70 and positive 30. So, basically, the resting membrane potential, that's the inner side. The inner side is negatively charged. So, the resting membrane potential is at negative 70 millivolts. And when it becomes active, or when when the action potential passes through that region in the membrane, it becomes the charge reverses, so it becomes positive 30. Okay, so basically during an action potential, the negative 70 becomes a positive 30 millivolt charge. Okay, and that is basically the activation, or basically that's the summary of your action potential. So, variations in ion concentration across the cell membrane allow neurons to generate this action potential, as I said. So, basically, the membranes, how does the membrane maintain that negative 70 and then it becomes positive 30? How does it happen when you have, when the ions cannot pass through the membrane? So, that membrane has channels embedded in the membrane. So, they are gateways in the channel. So you have sometimes basically there are some leaky membranes, so they sometimes allow some ions to pass through, but most of the time they are closed and they can only be opened by stimulus. So if you consider it, it's just like during say a convention, a lot of Comic-Con or anime convention, a lot of people outside, they wanted to come in. So some people have special VIP pass that can pass through. So that's the leaky. But when the gates open, basically people rushes in. So basically that's what happens during the movement of the ions in the membrane. So basically here is your membrane potential. So this is your, so as I said, you have two sides of the membrane. This is the extracellular side. You have an excess of positive charge here, mostly sodium ions, and then you have here your negatively charged intracellular side. So this is the cytoplasmic side, and this is your cell membrane. So this is hydrophobic part. They cannot easily pass through here. Now, this is just one region of your axon, for instance. So the action potential comes in from the dendrites towards the axon. So basically, the movement here is the moving positive charge, because remember, the resting potential is 70, negative 70. So at rest, when the neurons are not firing, so the charge here is negative 70. But when the action potential passes through, so the positive 30 charge is moving from, of course, the dendrites to the axon region of your neuron. So basically, this is the resting state. So how do they know the value of negative 70? Of course, they measured it. Okay, so you have an oscilloscope that measured the action potential. Actually, you did that. You will be doing that in your Labster activity. Okay, now the gates and channels control the flow of the ions. So how does the negative 70 becomes positive 30? Of course, they have gates and channels. There are several types. You have the voltage-gated channels, you have the ligand-gated channels, and the mechanically regulated channels. So for the voltage-gated channels, they respond to transmembrane voltage. So how does it happen? So remember, the potential, the positive 30 charge is moving from the hillock, the axon hillock, to the ends of the axon itself. So for example, the voltage gated channels around this region senses that the region on their neighboring region becomes positive 30, so they becomes activated again, they open. So again, basically it's like it's a relay. So when they sense that their neighbor is at positive 30, okay, so we will now open the channels, so we will now become positive 30. So that means the neighbor on my other side will sense that there's a change na, so it's time to open the channel. So basically that's the voltage gated channel. And then we have the ligand gated channels. The ligand gated channels are chemically regulated, so they respond to basically they are sometimes called receptors, because chemicals, actually neurotransmitters in particular, are binds to these channels for them to open. So usually these are found at the ends. So the synapse region, so the synapse is the region where the tip of the axon of the previous neuron is attached or connected. They're not physically connected, but they're very near each other. There's a space to the next cell. It can be a muscle cell, or it can also be another neuron. So basically pasapasahan. So the next one in the relay. Now, since the action potential, the charge is of course the electrical charge is passed through within the neuron, but since because of the synapse, you cannot have the electrical charge cannot actually jump from this across the synapse. So basically, when the potential reaches the tips of this axon, what happens is that it will trigger the release of the neurotransmitter, and that neurotransmitter will then be the one responsible for opening the channels on the next neuron, and so you will have the action potential continues on. Basically, remember, just like this one. For instance, this neuron is activated. So the signal here is electrical. So you have voltage-gated channels opening and closing across this axon. And then, when it reaches the tip, since the next neuron cannot sense the voltage of the previous one, because they are not really physically connected, instead, the tip of this neuron will release neurotransmitters that will be sensed by the next neuron. So if this is the next neuron, it will sense it and it will open the channel. So it will now generate that positive 30 electrical impulse that will then be propagated across the cell itself. So on and so forth. So basically, that's the general idea how the impulse travels across the cells, across the different neurons. And that is the ligand-gated channels. And then we also have mechanically regulated channels. These are usually found in the sensory neurons, usually the tactile sensory neurons, where we will basically the feeling of touch, because of course mechanically regulated, they are just like piezoelectric materials. So distortions in the shape induces the opening of these channels. So we have these mechanically regulated channels responding to distortions of the membrane surface. Now this is the anatomy, so-called anatomy of your channels. So these are voltage gated channels. So they have actually three ways, or you have they have three states: the open, the closed, the open, and the closed, the open, and basically you have inactivated. Okay, so why is it three? So the first one is closed. Basically, they are just closed. And then the opening channel, they allow the neurons to pass through. Now, the inactivated state is where you have here the gates. So the closed gate here is the reason why, even though it's, for example, for voltage gated channel, even though your channel senses a change in the voltage, the potential doesn't go back from the axon back to the cell body, but rather there's only one way of movement. That is because of the inactive state. So after the channel, for example, the normal state is closed, and the resting state, the channels are closed. When the action potential happens, it opens. When it's open, of course, it allows the ions to pass through. And then afterwards, they will become inactivated. Now, when they are inactive, it's basically the lock state. So no amount of stimulus can, in that state, no amount of stimulus can open this channel. Regardless of having attained the required voltage, it won't open until a certain time has passed. And that is the reason how the action potential is a one-way street. So basically, it only goes one way. So this is your ligand-gated channels. So here, you have spaces here for the neurotransmitters. So the ligands are the neurotransmitters. So you have your acetylcholine, for example, here. These acetylcholine, nicotinic acetylcholine receptors. So these are, once the ligand binds to your receptor, it opens and allows your ions to pass through. Okay, so neurons work through action potentials, as we said. So the action potential is a brief change in electrical condition at a neuron's membrane. So when a neuron fires, the charge differential across the neuron's membrane suddenly reverses polarity. Remember, it's negative 70, it becomes positive 30. So action potentials are all or nothing events, meaning it's either you have it or you don't have it. So they are basically binary. There is no halfway. There is no say negative 5 millivolts action potential. It's either you're negative 70 and then you become positive 30. So now, once the sodium channels are open, there are refractory periods in which another action potential cannot be generated in that neuron, and that is during that refractory period is basically the time when your channels are inactive. So they are basically locked. So they are not only closed, they are also locked. So if you consider them as a gate, they are not only closed, they are also locked. So we have several types. Absolute refractory period, that occurs when the sodium channels are inactivated and the potassium gates are open. And the relative refractory period occurs after the sodium channels are reset back to the rest and potassium gates are still open. So you have here your action potential. So it becomes negative 70, then positive 30. And then afterwards, it becomes back down to the resting state, negative 70. How does it do that? So basically, you have a sodium-potassium exchange pumps that restore the resting membrane potential. So the movement is 3 sodium for 2 potassiums. So that's the movement. So this is your action potential. So let's start with the resting state. The resting state, you have at -70, you have your positive sodium outside, the negatively charged proteins, and your potassium ions inside. Okay, so this is your plasma membrane. Now, you have a stimulus. So, it reached the threshold stimulus. So, this is the polarizing phase. So, the polarizing, because, it's called depolarizing because the membrane potential is polar. Why is it polar? Why is it considered polar? Because you have a very clear negative and a positive side. Now, the depolarization happens, because it reverses the polarity, so it becomes more positive. It actually approaches zero, so that no longer becomes polar, but actually it overshoots. It becomes positive 30. But anyway, it's still called depolarization. So depolarization, to the threshold, it opens the sodium channel and the sodium channel. And because you have a lot of sodium outside and a little sodium inside, of course, the tendency for the sodium is to come in. Okay? So it goes from -70 to the threshold, -55, and then it shoots up to positive 30. And then you have the repolarization phase. So at positive 30, channel inactivation gates closed, so your gates becomes locked, and your voltage-gated potassium channels open. So you have an outflow of potassium channels, and because remember, this time it becomes the inside becomes positive, so the potassium wants to go out. By the way, the channels are very specific to particular ions. If it's a sodium channel, only the sodium can pass, no potassium can pass. If it's a potassium channel, only the potassium can pass, not the sodium. So basically, that's the idea. So you have an outflow. In the repolarization phase, you have an outflow of positive channels. So why is it repolarization? So remember, at the start, you have a negatively charged cytosol. So the interior is negative charge. Once the sodium channel opens, the sodium ions comes in. So your cell membrane, the interior, becomes positive. It becomes positive 30. And since this is positive 30, once this potassium channel opens, of course, since this is positive, they want to get out. So the positive charges want to go out. And since the only positive charge that can go out is the potassium, so this is the repolarization phase. So you have an outflow of potassium. And then after that, you have a continuous repolarization until you reach back to the resting state. So potassium outflow restores the resting membrane potential. And then afterwards, the potassium channels will close. And then you have you now return to the resting state, but the ions are switched. And then that is where the sodium-potassium pump comes in. So sodium-potassium pump, it will restore the membrane potential by actually switching the different ions. So after the outflow of the potassium, it will the sodium potassium pump will work very hard to actually put sodium outside and potassium back inside the cell, and you go back to your resting potential. So basically, that's your action potential. So the action potential is moving from the axon hillock towards the ends of the axon. So basically, you have, for example, here, at the start, you have a current, you have your, remember, this is the resting potential. So once it opens, this area becomes positive, and then since the channels here are voltage gated, they will sense that there's a change in the polarity, so they will open again. So the movement is from left to right here in this diagram. So that's your membrane potential. Now the neurons are myelinated. So remember, you have myelin sheets or the Schwann cells producing myelin sheets in your membrane, in your neurons. Now what are these for? So they are like they're actually very much like remember how you will insulate wires. So basically, it's similar to that. They serve as insulation, and they also allows the movement of the action potential to go faster. Okay, so without this one, the potential will be very, very slow. Actually, there's a condition here. If the destruction of the myelin sheet causes paralysis. Okay, so you have movement here. Because why does wrapping the axon with myelin sheath helps the action potential to propagate faster? Because it actually produces nodes, and it concentrates your charge difference. So you have your basically a more concentrated movement of your charges. So the myelin is produced in the peripheral nervous system by the Schwann cells, and in the central nervous system by the oligodendrocytes. So you have a condition called multiple sclerosis. That is the degeneration of the myelin sheath, and depending on what neurons are being affected, if it's in the Schwann cell, in the peripheral neurons, the Schwann cells are being affected by the multiple sclerosis, the result is usually paralysis. But if it's in the central nervous system, it's usually dementia, mental retardation, etc., because it's the brain that's being affected. So here are your Schwann cells. So this is for it can be many neurons or one neuron at a time. So a single axon or a multiple axon. Okay? So synapses separate one neuron from another, and neurotransmitters bridge the gap. So as I was saying a while ago, so the action potential is electrical in nature across the cell, but between the two cells it becomes chemical. So the stimulus is transmitted from the previous neuron to the next neuron through the synapse. This is the space. The synapse, by the way, is just the space between the neurons. And in that synapse, that is where the neurotransmitters are released by the previous towards the next neuron for to activate the next neuron. So the presynaptic neuron, before, so the previous neuron, that is the one that releases the neurotransmitter, and the postsynaptic neuron binds the neurotransmitter with receptors on its membrane to stimulate signals. So this is where the receptor or the ligand-gated channels are usually located. So here is your synapse. So this is the previous neuron and then the next neuron. So here is the gap. So remember, this is the presynaptic neuron. You have an action potential passing through here. And then since, as I told you, they are not really touching, they have a space between. So this is the synaptic cleft. So once the action potential reaches this region, it will trigger the release of the transmitter. So in this region, there are many synaptic vesicles on standby. So the neurotransmitter are already produced, and they are just waiting for the signal. They are waiting for standby. So once the signal occurs, that is the electrical charge difference in the membrane. So it will now, this synaptic vesicle will then be bind to your membrane and release your neurotransmitters. And then these are the ligand-gated channels. So they will bind to the ligand-gated channels, and they will open the sodium channels. It will start na the action potential on the postsynaptic neuron. Okay, so we have several types of neurotransmitters here, depending on what type of cells are producing them, and what they are for. So graded response create fine neural control. So we also have a graded, although the firing of the action potential is all or nothing. We have recent studies are showing that you can have some sort of a graded response. Basically, graded potentials start the signaling process in the dendrites and cell body. So we have hyperpolarizing graded potential. So they are also called the inhibitory postsynaptic potential. So it causes the membrane to become more negative, making it harder to stimulate the action potential. So that means you will require a greater or a larger amount of stimulus. So this is more on activation of the actual neuron, not in the action potential side where the voltage gated channels are involved. So this is usually where your ligand-gated channels are involved. Now, how does it do? Remember, if you are a coffee drinker, when you drink coffee, you tend to drink coffee 1 cup a day, and then you try to up your dose, or eventually, even 1 cup doesn't make it, so you need to up your dosage of coffee, so on and so forth. So that is basically a way of your body to actually not exactly adapt, but yeah, they are adapting to the excess of the stimulants by actually requiring you to intake more and more amount of stimulants for the same amount of response to happen. So basically, this is somewhat similar to this one, hyperpolarizing graded potential type, the inhibitory postsynaptic potential. Now, for the depolarizing graded potential or the excitatory postsynaptic potential causes the membrane to become less negative, making it easier to stimulate an action potential. So this one is the opposite. So if the hyperpolarizing graded potential makes us require a more intense stimulus to actually be stimulated. I mean, for example, you need to get more and more or higher and higher dosage of caffeine for you to wake up properly. For the depolarizing graded potential here, it allows the person to become much more sensitive to something. So, for example, if you work in a quality control center or a QA in a QA laboratory, sometimes your sense of smell or your sense of taste, for example, if it's a food manufacturing company, your sense of taste becomes very fine-tuned to very, very slight changes in the taste of the product if you are always taste testing it. So that's what the polarizing graded potential is like for the neurons. So just a small amount of stimulus can activate your potential. Okay, so that's it for this section. So in the next section, we'll discuss about the structure of the meninges and the brain structure.